Today, let us dive into skeletal muscle contractions, or what we can call sliding filament theory. And in this session, we are going to look at the brief structure of a functional unit of a skeletal muscle, which is known as sarcomere. Then we are going to look at how impulses transmit across the neuromuscular junction. Then we shall finish with the sliding filament theory of muscle contraction. And as I begin, if you are new on this channel, please subscribe. Go to the subscription button and subscribe. Then you can like the video, then share to many others so that they can continue to learn with us. So, skeletal muscle contraction, it occurs in the functional unit of a muscle known as the sarcomere. So the sarcomere is the functional unit of the skeletal muscle, and it is striated. The sarcomere is striated, containing the thin actin and the thick myosin. Thin actin and thick myosin. And by definition, what is a sarcomere? The sarcomere is the length. If this is the Z line or the Z disc, and this is the Z disc or the Z line, the distance between two Z lines is what we refer to as the sarcomere. So the sarcomere is the distance between two Z lines. And this sarcomere, which contains the thin actin and the thick myosin, so this is the actin filament, whereby then this one becomes the myosin, and this myosin has what we call the myosin head. It has the myosin head. Still, this is actin, thin actin, then this is myosin, which is thick. And the center line is what we know as the M line. It has the M line, and the zone in between, we refer it to as H zone. This is the H zone. And then the zone where the actin overlaps with the myosin, together with the myosin head, is known as the A band. You can call it A band or dark band. Then the zone or the distance where the actin is not overlapping with the myosin, we call it light band. You can call it light band or I band. So, meaning even this zone from here, from the Z line to where the myosin is overlapping with actin, still this one becomes the A band, the I band. This is the I band. Then this one is the A band. Then this one in the middle is what we are calling the H zone. And during muscle contraction, the actin filaments, which are thin, they move past the stationary myosin. So the myosin does not move. It is stationary. The thick myosin remains stationary, whereby the actin filaments slide past the stationary myosin. That is during muscle contractions. And during muscle contractions, because of the sliding of the thin actin past the stationary myosin, you're going to see the Z lines come closer. Then the I band shortens. That is during muscle contraction, as we shall see after sliding filament. So this is the brief structure of a sarcomere. After looking at this structure of a sarcomere, the next thing we are looking at transmission of impulses across the neuromuscular junction. Transmission of impulse across the neuromuscular junction. What is a neuromuscular junction? it is a junction, or it is a meeting point where the neuron meets the skeletal muscle. And the membrane of the skeletal muscle is known as the sarcomere. Then the sarcomeres are within the muscle membrane. So this membrane is known as the sarcomere. Then this is the membrane of the neuron, and this neuron contains the synaptic vesicle with neurotransmitter acetylcholine. So the neurotransmitter is acetylcholine at the neuromuscular junction. Excuse me. And the receptors of acetylcholine at the sarcomere, we are going to call them, these are acetylcholine receptors. So when it is released, we are going to see how does it bind. So in case you touch in fire or you have stepped on a nail, the sensory receptors are going to send impulses to the neuron, and when the impulses arrive of the impulse in the neuron, remember at rest the outside is positively charged, inside is negatively charged. But upon arrival of an impulse, it is going to cause influx of sodium ions. It is causing entry of sodium ions inside the neuron, resulting into the inside of the neuron to become positively charged and outside negatively charged. And this is what we know as depolarization. So the neuron becomes depolarized, undergoes depolarization, whereby the inside becomes positive and outside becomes negative due to influx of sodium ions. And this inside of the cell of the neuron becoming positively charged and outside negatively charged, it is going to stimulate the calcium gated channels. These are calcium high voltage gated channel. It is going to open, causing influx of calcium into the neuron. And when calcium enters into the neuron from the NMJ, it is going to create what we call a power stroke. And this power stroke is going to bring about contractions within the neuron, causing this vesicle to move, to come and fuse with the membrane. So this vesicle comes and fuses with the membrane, and after fusing with the membrane of the neuron, it is going to release the neurotransmitter acetylcholine by exocytosis. So this release is known as exocytosis. So the neurotransmitters are released by exocytosis in the presence of calcium, which causes contractions, causing the membrane, the vesicle to fuse with the membrane, hence releasing the neurotransmitter. And after releasing this acetylcholine, it comes and attaches to its receptors on the sarcomere. And after touching on its receptors on the sarcomere, it is going to increase the permeability. Remember, in the junction, we have there sodium ions. So these sodium ions, upon the binding of acetylcholine on its receptors on the sarcomere, is going to cause influx of sodium ions. So sodium ions which were in the junction are going to move inside the cell, and influx of sodium ions inside the muscle is going to cause them inside of the muscle to become positively charged. That is what we call depolarization. And after being depolarized, when it sums up to reach a threshold value, it is going to fire an action potential. An action potential is going to be fired, which we abbreviate as AP. And what it transmits or causes propagation of the action potential is what we call the transverse tubules. We have the transverse tubules, or you can call them the T tubules. are the one that causes propagation of an action potential across a muscle cell. And remember, we have the transverse, we have also the aerotuberous. Aerotuberous, we call them longitudinal tuberous. So as an action potential is propagating down the muscle cell, it is going to shake the aerotuberous, what we are calling the longitudinal tuberous. And these longitudinal tuberous are attached to the cisterns, the cistern containing within the sarcoplasmic reticulum. Sarcoplasmic reticulum. And this sarcoplasmic reticulum is rich with calcium ions on the cisterns. So the propagation of an action potential by the transverse or the T-tuberous is going to shake the longitudinal tuberous, causing the release of calcium ions from the cistern. And when these calcium ions are released from the cistern within the sarcoplasmic reticulum, they are going to be released in the membrane. So we are seeing release of calcium ions from the sarcoplasmic reticulum into the plasma membrane of the muscle. So calcium ions are released, and what has caused the release is the action potential being propagated by the T-tuberous, which shake the aerotuberous, leading to the release of calcium ions into the membrane. And when these calcium ions are released, they come and attach on the troponin. This is what we call troponin. And the troponin is composed of two different isoenzymes. We have troponin C, troponin T, and troponin I. So when calcium is released into the membrane, increased levels or high concentration of calcium released within the membrane is going to bind with the troponin protein, especially at the troponin C. And when it binds to troponin C, remember troponin is on the actin. This is the actin filament. This is the actin filament. So when troponin binds, this actin filament at a relaxed state, the binding site of on the actin, they are always covered by tropomyosin. So at a resting state, the binding sites of this are always covered by tropomyosin, so that the myosin head does not bind. So, and also this one is also covered by tropomyosin. So that is at a relaxed state. But when there is depolarization of the muscle, leading to propagation of an action potential by the transverse tuberous, causing release of calcium ions from the sarcoplasmic reticulum, increased levels of calcium ions binds to troponin, and then troponin is also binding to the tropomyosin. And what happens? This tropomyosin, the troponin binding to calcium also binds to tropomyosin, causing a complex known as tropomyosin complex. And this tropomyosin complex formed after calcium binding, to troponin C. It leads to tropomyosin complex, which removes and exposes the binding site on the actin. And the binding site on the actin they are known as F-actin sites. And why is tropomyosin always covering them? Is to prevent myosin head binding to them when there is no trigger of contraction. But when the contraction comes, the release of calcium ions from the sarcoplasmic reticulum binds to troponin C, forming tropomyosin complex, which removes the tropomyosin to expose the F-actin binding sites on the actin filament. So now at this step, the F-actin binding sites are exposed, so the myosin head can easily bind. So this is the myosin, and this is the myosin head in the presence of ATP, energy provided by ATP. It is going to cause the myosin head to bind to the F-actin. So if here there is the F-actin on the actin filament, in the presence of ATP, if this is the myosin head, it is going to come and bind after removing the tropomyosin, which was masking the binding site. So when I remove the tropomyosin, now the myosin head is able to recognize the F-actin binding site on the actin filament and comes and binds. Then in the presence of ATP, the energy is provided by ATP, and this same ATP, after myosin head binding on the F-actin, it is going to cause the power stroke, that is the bending. So when the myosin head binds, after binding, it causes the power stroke provided by energy in ATP, and then that binding and forming the stroke is pulling the actin. It pulls the actin filament to move along. If it binds, then it causes the power stroke, it pulls. That's why we are seeing that this ATP, after the myosin head binding, it forms what we call the power stroke. And this power stroke causes the bending of the myosin head. So you can see this myosin head was straight, but when it binds on the F-actin site on the actin filament, it bends. And when it bends, it pulls along, or it moves along with the filament, with the actin filament. And then at that end, we are going to resynthesize, we are going to release ADP, which is going to be used whereby ADP will bind again with the phosphate inorganic to restore ATP. And this ATP, so we are seeing when F-actin sites are exposed, the myosin head keeps binding on F-actin, causing a power stroke, removes. We resynthesize ATP. ATP comes back, and then causes another one to bind, causes a power stroke, pulls. The same thing comes back, binds, pulls. That is what we are calling sliding filament theory. Every time the myosin head binds, causes power stroke, pulls along. Binds, causes power stroke, pulls along. Resynthesizes ATP like that, and this is what we call sliding filament theory. And at the end, or in summary, what we have already covered, we have seen arrival, point number one, we have seen that arrival of impulse in the neuron causes depolarization due to influx of sodium ions, and this depolarization causes influx of calcium ions by opening calcium high voltage gated channel, and this influx of calcium ion causes contraction in the neuron, leading to the fusion contractions, and this contraction brings about fusion of the synaptic vesicle with the membrane with the membrane, leading to release of acetylcholine neurotransmitter by exocytosis. And when acetylcholine is released at the neuromuscular junction, it binds on the receptors the acetylcholine released binds on sarcomere receptors, causing depolarization due to influx of sodium ions. And when depolarization sums up to reach a threshold intensity, it results into formation of an action potential, and this action potential formed is propagated via the T tuberous, what we call the transverse tuberous. And the propagation of an action potential via the transverse tuberous are going to cause the release of calcium from sarcoplasmic reticulum into the plasma membrane of the muscle. That is what happens after the release of calcium ions. After the release of calcium ions into the plasma membrane of the skeletal muscle, the calcium binds, the increased levels of calcium goes and binds on the troponin, especially C, excuse me, leading to formation of troponin tropomyosin complex, and this complex is the one that removes tropomyosin to expose F-actin binding site. And when they are exposed, the myosin head come and binds in presence of ATP, as we have seen, and in presence of ATP, after binding, it creates a power stroke. And this power stroke, it causes the actin to slide past the stationary myosin. It keeps binding, forming a power stroke, pulling. Binds, power stroke, pulls. And that is what we call muscle contraction. So if I can illustrate it here, if the myosin head binds on the actin, on F-actin side, it pulls. Even this one binds, pulls. So as they are pulling, what is going to happen? What is going to happen on the sarcomere is that, so as they are binding, pulling along the stationary myosin, what is going to happen to the sarcomere? Sarcomere will shorten. So the sarcomere length shortens. Why is it shortening? Because we are pulling the Z line. The actin, as they are sliding over, they are pulling the Z line. So the sarcomere shortens. Then after shortening, even the light band shortens. The light band also shortens. Then the H zone also shortens. The H zone shortens. Then the A band increases. The A band increases. That is what will happen. And if it were to illustrate it here, the contracted muscle, if we have the M line, we have the Z line, this is another Z line. So we are seeing during contraction, We have our myosin, which for them remain stationary with the head, the myosin head. So what we are saying is that you can appreciate that the H zone decreases, and even the light band decreases. So this one is the light band. Then you can appreciate the darker band increases. Then the light band, what you are calling the I band, decreases, and the sarcomere shortens compared to this. That is during contractions. And this is what happens during muscle contractions. And after the muscle contracting, we are going to see the calcium which was released is going to be transported by the ATP back into the sarcoplasmic reticulum. And when calcium goes back, there will be no longer contractions. And then the muscle will be able to relax again. So in summary, what we have looked at, we have seen that arrival of impulse causing the neuron to become depolarized, and depolarization causing opening of the high voltage calcium gated channels, causing contractions inside the neuron. Hence, the vesicle comes and fuses with the membrane, leading to the release of neurotransmitter by exocytosis. Then this neurotransmitter binds on its receptors on the sarcolemma, causing depolarization. Then the depolarization can bring about an action potential, which is propagated by the transverse tuberous. Then these action potential are going to shake the systems attached on the aero tuberous, causing release of calcium from sarcoplasmic reticulum into the plasma membrane. And when it is released into the endoplasmic membrane, it is going to go and bind to the troponin, causing formation of troponin tropomyosin complex, which is going to remove the tropomyosin which was hiding the active sites from the actin, and after being exposed, the myosin head comes and binds in the presence of ATP, causing the power stroke, hence causing contractions of the muscle. Calcium binds, form troponin tropomyosin complex, removes the tropomyosin, myosin head binds, the muscle contracts. Like that. Then it is due to the power stroke, and we have seen at the end of this contraction, what happens to the sarcomere, it shortens the length, the light band shortens, the H zone shortens, the A band increases. So this is what we can discuss for you under muscle contraction, what we call sliding filament theory. Hope you have learned a lot from us. Always be tuned on. Please subscribe so that you are always notified whenever we release videos. Thank you so much.